Microbial ecology, macrobiomy, clinical and sanitary microbiology, lecturer Andrushkova Natalia Grigorievna. Ecological macrobiology. Biosphere of the Earth was formed about 3 billion years ago. At that point of time, the only inhabitants of the Earth were prokaryote organisms, bacteria which played a big role in its creation. Nowadays, the total mass of microorganisms of the planet is more than 7 billion tons. Protozoa and algae, all plants, animals. Moreover, the enzymatic activity of bacteria biomass in many times exceeds this process in plants and animals. Such wide distribution of microorganisms, their great participation in the deep decomposition of various organic compounds, causes their colossal role in biogeochemical cycles, rotation of substances and energy in nature that originated in the early stages of geological history of the Earth and are continuing now. Ecological macrobiology studies relations in micropopulation and influence of microbes that are exist together in certain biotope to each other. Ecology from Greek oikos, home, place or residence. Science about the formation and functioning of biological systems, their relationship with the environment. Microbial ecology is a science about the places where microorganisms are populated and their ecological interrelations. When studying microbial ecology, the same concepts as in the general ecology are used. Population is a collection of individuals of the same species that have the same gene pool and live in a particular biotope. Biotope is the place of residence of the population, the site which homogeneous conditions of existence, for example skin, stomach, small or large intestine. The term ecological niche defines as a rule not only the place of a microorganism in space, but also its functional role in a microbiocynosis. In the macrobial world, this is primarily due to the nutritional needs of bacteria, the way of reproduction. Microbiocynosis is a totality of populations of different microorganisms living in the same biotope. For example, the oral cavity vagina. Ecosystem, ecological system. A biological system with a functional unity of microbiocynosis and the environment. Its structure includes both biotic and abiotic components. Biotic components are represented by a set of populations and bacterial community that form microbiocynosis. Abiotic components are the numerous physical and chemical conditions for the existence in ecological system. The size of ecosystem can be different, a large or small intestine, a separate pond or swamp, or the like. As Mr. Vinogradsky suggested, the bacteria that form ecological systems are usually divided into autochthonous and alloctонous. Alloctонous microbes are typical inhabitants of the microbiocynosis and are always present there. Nitrophine bacteria in the soil, lactobacilli, and bifidobacteria in the intestinal tract, epidermidis or the skin surface. This is due to the presence of certain nutrients that are characteristic for the ecological system. Microbes which presence depends on the influence of certain factors, for example changes in the concentration of substances in the ecosystem, are called alloctонous bacteria. They are temporary residents of microbiocynosis. General types of microbial interrelationships are symbiosis, antagonism, predation, and mutualism. Symbiosis a situation in which two organisms live together in a close partnership. It consists of mutualism, commensalism, and parasitism. Symbiosis, in the narrow meaning of this word, is one of the forms of interpopulational relationships, in which both are associated with a certain benefit. For example, the coexistence of fungi and algae in lichens. In the microbial kingdom, the relationships between the normal microbiota and the host. Mutualism is a mutually beneficial cohabitation when the existence of populations is interrelated. Commensalism is a form of symbiosis in which populations live using the host, but do not harm it. This type of interaction is common to most bacteria, which are the normal microbiota of the body. A classic commensal interaction between microorganisms called satellitism arises when one member provides nutritional or protective factors to the other. One example of nutritional satellitism is observed when one microbe provides a growth factor that another one needs. In this example on blood agar, Staphylococcus aureus provides growth factors of Haemophilus influenzae, which grows as tiny satellite colonies near the streak of Staphylococcus. By itself, Haemophilus could not grow on blood agar. Parasitism is a widespread form of ecological interrelations in which one of the species of bacteria uses a more organized organisms, as example human, animal, or plants, as a habitat for the realization of its own nutritional needs. Such microbes are pathogenic and act as causative agents of infectious diseases. Synergism is an interrelationship between two or more free-living organisms that benefits them but is not necessary for their survival. Together, the participants cooperate to produce a result that none of them could do alone. In synergism, two organisms that are usually interdependent cooperate to break down a nutrient neither one could have metabolized alone. Predation. A predator is a form of consumer that actively seeks out and ingests live prey. And here you see illustration for microbial predation. In the antagonistic or competitive type of interaction, the phenomenon of suppression of the vital activity of one population by another is observed. Such microbes produced bacteriocins, antibiotics, fatty acids that cause the death of other populations or restrain their reproduction. This mechanism underlies the colonization resistance of normal microbiota. Antagonism or amensalism is a form interaction between organisms when one microorganism inhibits development of others. Mechanisms of antagonism are competition for nutrient substrate, different spread of growth, excretion of acids, alcohols, ammonia by microorganisms antagonist, excretion antibiotics, bacteriocins by microorganisms antagonist, and predation. Microbes that adapted to the body and normally live on the skin, in the alimentary tract, and in other sites are called the normal resident microbial flora, or indigenous flora. The development of medical microbiology is inextricably linked with the deepening of knowledge about the role of normal microbiota in the functioning of the human organism. In the process of evolution, a mutually beneficial symbiosis of a macroorganism with certain kinds of friendly microorganisms was formed. Skin, mouth, nasopharynx, stomach, intestines, urogenital tract have their own specific microbiota. All these local microbial cyanosis closely interact with each other and with the macroorganism, forming a single microecosystem. The active process of forming a symbiosis of the human body with physiological bacteria begins from the moment of its birth, and of the success of this stage, which can last up to 2 years, depends on the state of the microbial ecological system throughout the entire subsequent life of the individual. The healthy role of normal microbiota in human life was first identified at the beginning of the 20th century by our outstanding compatriot, Nobel Prize winner Mechnikov. In his famous monographs Studies of Human Nature and Studies of Optimism, he outlined the conceptual view on the relationship of the macroorganism and its microbiota, substantiated the importance for human health of symbiosis with physiological bacteria, and laid theoretical foundations for the development of bacterial prophylaxis methods and bacterial therapy. The role of normal flora in human organism. Antagonism, inhibition of growth of potentially pathogenic microorganisms, stimulation of the immune system, producing of essential nutrients as vitamins, as example vitamin K, vitamin E, and vitamin B, enzymes. Normal flora of the digestive tract are essential for degrading cellulose. The human microbial ecology studies the composition and functions of the natural microbiota of various human biotopes, the relationship of endogenous microbiota to the macroorganism and the environment, as well as the relationship between individual components of microbial biocyanosis in normal condition and in pathology. In humans, the organism for the first time comes into direct contact with microorganisms, first with the microbiota of the mother's birth canal, and then with the microbial world of the environment. The result of this contact is the formation of a new ecological system consisting of a macroorganism and numerous microbial populations associated with epithelial biofilms of mucous membranes, of non-sterile cavity organs, and human skin. The microbial ecological system of human is a complicated dynamic complex, formed in the process of onto and phylogenesis. It includes a macroorganism and a diverse by quantitative composition and taxonomic affiliation association of bacteria, viruses, protozoa, as example microbiota or microbiomes, as well as their metabolites concentrated in relatively open biological systems of the organism. Microbial populations occupy all surfaces of the human body, which are directly or indirectly in contact with external environment. As example, oral cavity, esophagus, stomach, small and large intestine, vagina, nasal passages, skin, upper and lower respiratory tract. The departments of the human body populated by microorganisms are called biotopes. The totality of microbial populations living in the same biotope is united under the term microbiocenosis. The concept of normal microbiota combines a multi-species complex of microorganisms inhabiting human biotopes under normal physiological conditions. Normal microbiota should be considered as an integral, vital part of the human body, a kind of microbial multifunctional organ that performs independently and in cooperation with other organs and systems of the macroorganism, a variety of physiological functions that ensure the homeostatic state of the organism as a whole. In the biological systems biotopes of the human body, numerous microbial populations of normal microbiota are concentrated in a specific integral structure, the epithelial biofilm. On this slide you see sites that harbor a normal flora. Skin and its contiguous mucous membranes, upper respiratory tract, gastrointestinal tract, various parts, outer opening of urethra, external genitalia, vagina, external ear canal, external eye, lids, conjunctiva. Opposite sterile microbe-free sites and fluids. It consists all internal tissues and organs like heart and circulatory system, brain and spinal cord, bones, muscles, lungs, kidneys and bladder, liver, and blood, urine in kidneys, ureters, bladder, cerebrospinal fluid, semen prior to entering the urethra, amniotic fluid surrounding the embryo and fetus. The human integuments are in constant direct contact with the external environment, including the exogenous microbiota. The outer layer of the skin is represented by a strong multilayer keratinizing epithelium, epidermis. Low moisture content and bactericidal action of the secretions of the skin glands prevents excessive reproduction of microorganisms. The epidermis is waterproof and counteracts damaging mechanical factors and interferes with the translocation of microorganisms. Ordinarily, there are two cutaneous populations. The transient population or exposed flora clings to the skin surface but does not ordinarily grow there. It is acquired by routine contact and it varies markedly from person to person and over time. The transient flora includes any microbe a person has picked up, often species that do not ordinarily live on the body, and it is greatly influenced by the hygiene of the individual. The resident population lives and multiplies in deeper layers of the epidermis and in glands and follicles. The composition of the resident flora is more stable, predictable, and less influenced by hygiene than is the transient flora. The normal skin residents consist primarily of bacteria and yeasts. The main microbes that inhabit the skin are Staphylococcus, Micrococcus, Corynebacterium, Candida albicans, Neisseria, Peptostreptococcus, Propionibacterium. This slide illustrates model of colonization of intestinal tract. This table presents the quantitative characteristics of the microbiota of various biotopes of the gastrointestinal tract. Urogenital tract. Vaginal biocenosis contains about 10% of the microorganisms of a woman's organism and plays a leading role in maintaining her health. Normal vaginal biocenosis of a woman is a specific, vitally important organ that performs a very wide range of protective, metabolic, regulatory, and other functions. The vagina is covered with a multi-layered flat epithelium which undergoes cyclic changes in response to the action of sex hormones. Estrogen hormones contribute to the saturation of the vaginal epithelium with the glycogen, which is utilized with the vaginal biovariants of the saccharolytic bacteria of the Doderlein group. With the development of these microorganisms, a significant amount of lactic acid is formed. Acidic medium with a pH of 4 to 5 prevents the biotope from being colonized by acid-sensitive microorganisms. Therefore, the species composition of the biocenosis is rather monotonous. The main place in it is occupied by lactic acid bacteria, up to 97% of the total number of microorganisms. The share of all the other numerous representatives of the vaginal biocenosis does not normally exceed from 3 to 4%. Since their first description in 1892 of lactobacilli as the predominant microbial of the normal vaginal biocenosis, their significance in maintaining the colonization resistance of the female urogenital tract remains unchallenged. It is known that the first signs of vaginal dysbiotic disorders are a decrease in the concentration of anaerobic sucrose-containing flora or the loss of its biological protectees. Genital tract consists Lactobacillus candida albicans, Corynebacterium, Enterobacteriaceae, Micrococcus, Staphylococcus epidermidis, and Streptococcus. On this slide you see photos with norm and pathology vaginal microflora. Norm, candidosis, and various microflora. The state of dynamic equilibrium between the human organism, its microbiota, and the environment, in which human health is at an optimal level, is commonly called eubiosis. Eubiosis, normobiosis, is characterized by a stable composition of microbiocenosis and a full volume of their physiological functions. The state of eubiosis is the manifestation of a harmonious symbiosis of the macroorganism with its natural symbiotic microbiota, reflecting the motile equilibrium between them. There are a huge number of factors affecting the composition and functional properties of individual microbiocenosis. These include, for example, the genetic characteristics of each individual, peculiarities of the formation of the primary biocenosis in the neonatal period, the age of the individual, climatic and geographical conditions of residence, living conditions, food traditions, etc. Disorders of eubiosis, manifesting by the development of microecological disorders in various biotopes, which are associated with changes in the normal composition and functions of endogenous microbiota, are commonly called dysbiosis. In modern conditions, microecological abnormalities are observed in patients of practically all clinics and hospitals. The dysbacteriosis is a stable disturbance in species composition of normal human microflora in certain organ which is accompanied by clinical signs and demands specific correction. Modern methods of correction of disturbances in the human microbial ecosystem are based on the use of a wide range of biological preparations. Aerobiotic is suspension of live bacteria that able to renew number of missed or deficient species for correction of normal flora. Main effects of aerobiotics: replacing bacteriotherapy, antagonistic activity, detoxification of substances, immunostimulation, synthesis of vitamins, improving of gastrointestinal tract function, inactivation of cancerogens. Clinical microbiology as a separate field of medical microbiology has been formed during the last decades. This was facilitated by the spread of infectious diseases and numerous purulent and septic complications in non-infectious hospitals and departments of various profiles. Constant evolution of pathogens, especially of the diseases caused by them, and ratio of their increase led to an increase in the medical and social significance of nosocomial, hospital-acquired infections for the healthcare system, initiated a review of the approaches to solving problems associated with this situation. Increase in the incidence of nosocomial infections is determined by a number of reasons, including an increase in the number of people of high-risk groups, introduction of complex surgical interventions, minimally invasive technologies, other instrumental diagnostic methods. It should be noted that uncontrolled irrational use of antibiotics in fight against infectious processes led to the selection and further widespread distribution of antibiotic-resistant strains of microorganisms. It also causes changes in normal microbiocenosis of the skin, mucous membranes, and intestine. The appearance of hospital strains, especially hospital ecological variants of pathogens with multiple drug resistance, increased resistance to the action of environmental factors, various disinfectants and antiseptics. The main objects of clinical microbiology study opportunistic microbes and their relation with human organism and human population, nosocomial infections, normal microflora and dysbacteriosis, control for sanitary epidemiological conditions in hospitals. Causing disease by normal flora microbes. Because the normal flora and their body are in a constant state of change, the symbiotic relationships are not absolute, and a commensal can convert a parasite by invading body tissues and causing disease. Members of the flora can also become infectious if the health of the host is compromised, as by cancer or AIDS, allowing opportunists, such as the agents of pneumococcal and pneumocytic pneumonia, to overcome the weakened host defenses. Under corresponding circumstances, the normal microflora can cause endogenous infection, unusual location, and immunodeficiency. Groups of pathogenic microbes. Pathogenic microbes have been traditionally divided into two categories, depending upon the nature of the microbe-host relationship. Examples include the influenza virus, plaguebacillus, rabian virus, malarial protozoa, and other microorganisms with well-developed qualities or virulence. Primary pathogens are generally associated with a distinct, recognizable disease. The degree of pathogenicity of true pathogens ranges from weak to potent. For instance, infection with the cold virus is mild and non-fatal, whereas infection with the rabies virus is nearly 100% fatal. Opportunists are not considered pathogenic to the normal, healthy person and do not usually have well-developed virulence properties. True pathogens or primary pathogens are capable of causing infection and disease in healthy persons with normal immune defenses. Influenza virus, plagiobacillus, rabies virus, malarial protozoan. Opportunistic pathogens cause disease when the host's defenses are compromised or when they become established in a part of the body that is not natural to them. Conditionally pathogenic microorganisms include a large group of bacteria, viruses, fungi, protozoa, which belong to different taxons. Under certain conditions, they can cause numerous inflammatory diseases in humans and complicate the course of the main pathological process. Features of opportunists. They are not intracellular parasites. The main virulence factor is endotoxin. Release great quantity of exoenzymes, as example hyaluronidase, coagulase, neuraminidase, lecitinase, nuclease, fibrinolysin, others. Set of virulence factors is different for different strains. Virulence factors have low specificity. The main genera that contained opportunistic members: bacteria, Streptococcus, Peptostreptococcus, Escherichia, Enterobacter, and others. Fungi, Candida, Cryptococcus, Pneumocista. Viruses, two types of herpes simplex, varicella virus, papillomavirus, adenovirus, and enterovirus. Opportunistic infections, from Latin opportunus, means convenient and profitable, is a group of diseases caused by opportunistic or conditionally pathogenic microorganisms. Their development is provided by three main factors of the epidemic process: the biological properties of the pathogen, the state of microorganism, reduced reactivity, the presence of immunodeficiency, damaged skin and mucous membranes, and certain environmental conditions. Populations of microorganisms that cause such diseases are heterogeneous in their biological properties, in particular pathogenicity, resistance to antibiotics. They are characterized by a high infectious dose, which causes the onset of the infectious process. The influence of external environment is associated with the presence of certain factors of transmission of pathogens in the human population. Pathogens of opportunistic infections spread very quickly in hospitals, endangering large groups of patients. Opportunistic infections, infections caused by opportunistics, normal flora microbes, transient bacteria in immunocompromised hosts. Immunocompromised patients are those patients whose immune mechanisms are deficient because of immunologic disorders, chronic diseases, or immunosuppressive therapy. Immunocompromised patients who are identified as high-risk patients have the greatest risk of infection caused by airborne or waterborne microorganisms. Opportunistic infections result from at least one of three basic mechanisms. First, exogenous acquisition of a particularly virulent pathogen. Second, reactivation of an endogenous latent organism. And last, endogenous invasion of a normally commensal or saprophytic organism. Nosocomial infections are any bacterial, viral, fungal, or parasitic diseases of patients and medical personnel whose infection occurs during provision of medical assistance in healthcare and medical institutions. During the main disease, these infections significantly worsen its course. drastically complicate laboratory diagnostics and treatment, and also complicate the prognosis and consequences of the disease, which sometimes leads to death of the patient. Any infection acquired during hospitalization. Nosocomial infection acquired in a hospital or healthcare facility. An onset of symptom more than 48 hours after admission, shorter hospital stays, may not be recognized until after discharge. Hospital-acquired infection (HAI), also known as a nosocomial infection, is an infection that is acquired in a hospital or other health care facility. To emphasize both hospital and non-hospital settings, it is sometimes instead called a healthcare associated infection. The most common nosocomial infections involve the urinary tract, the respiratory tract, and surgical incisions. Bacteria that account for the majority of nosocomial infections. Gram-negative intestinal flora are cultured in more than half of patients with nosocomial infections. Gram-positive bacteria and yeasts make up most of the remainder. True pathogens such as Mycobacterium tuberculosis, Salmonella, hepatitis B, and influenza virus can be transmitted in the clinical settings as well. There are two types of ways of infection. Exogenous, in result from diagnostic and therapeutic procedures. Catheterization of the bladder or a blood vessel injection, when surgery procedure, in result of contact with medical personnel, other patients, visitors, from contaminated air or water. Endogenous activation of resident microflora in immunocompromised human. Main routes of transmission include contact transmission, droplet transmission, airborne transmission, common venereal transmission, and vector-borne transmission. Contact transmission, as description by direct contact, the most important and frequent or indirect contact with the contaminated instruments, needles, or dressings. Droplet transmission, when droplets containing microbes from the infected person are propelled a short distance through the air and deposited on the patient's body by coughing, sneezing, talking during the bronchoscopy. Airborne transmission. Dissemination can be either airborne droplet nuclei, small participle residue, or evaporated droplets containing microorganisms that remain suspended in the air for long periods of time, or dust particles containing the infectious agents. Legionella, Mycobacterium tuberculosis, and the rubeola and varicella viruses. Common venereal transmission by contaminated items, food, water, medications, devices, and equipment. Vector-borne transmission by vectors, mosquitoes, flies, rats, and other. Feature of hospital infections, causative agent, adaptation to hospital condition, high level of genome changeableness, intensive exchange of genome material, high level of microorganisms virulence, resistance of microbial strains to a lot of antibiotics and disinfectants. The most common agents of nosocomial infection. Gram-positive cocci, Staphylococcus aureus, Staphylococcus epidermidis, and Staphylococcus saprophyticus, Streptococcus pyogenes, Streptococcus viridans, and Streptococcus pneumoniae. Gram-negative rods. Proteus vulgaris, Proteus mirabilis, Escherichia coli, Serratia, Salmonella, Pseudomonas, Klebsiella pneumoniae, Klebsiella ozonaya, and others. Obligate anaerobes, gram-negative rods, bacteroids and Fusobacterium, gram-positive cocci, Peptostreptococcus, gram-positive spore-forming rods, different types of Clostridium. The emergence and spread of antimicrobial resistant bacteria (ARB) is one of the most serious public health threats. Bacteria such as vancomycin-resistant enterococci (VRE) and glycopeptide intermediate-sensitive Staphylococcus aureus present hospitals with the prospect of a post-antibiotic era, in which few if any therapeutic antimicrobial agents remain effective. Compared to infections caused by susceptible strains, infections caused by antibiotic-resistant organisms are more likely to prolong hospitalization, to increase the risk of death, and to require treatment with more toxic or more expensive antibiotics. A high percentage of hospital-acquired infections are caused by highly resistant bacteria such as methicillin-resistant Staphylococcus aureus (MRSA) and vancomycin-resistant enterococci. Sanitary microbiology: science of the microbiota of the environment, water, air or soil, and related processes which can adversely affect human health. The subject of study in sanitary microbiology is the microbiota of the environment and its effect on human health. Now, sanitary microbiology is an independent science, standing at the joint of microbiology, hygiene, epidemiology, immunology, chemistry, computer technologies, and other sciences, but is equally necessary for specialists of various profiles. The beginning of the development of sanitary microbiology can be considered in 1888. The French physician Mr. Mace suggested examining the coli as an indicator of fecal contamination of water. Task of sanitary microbiology. First, development, improvement, evaluation of microbiological methods of environmental object research. Assessment of the ways in which humans and animals affect the environment. This problem is significant to sanitary microbiologists due to the fact that both humans and animals are the source of environmental contamination by both pathogenic microorganisms and other microbes, which can adversely affect the objects of the environment. Third, study of conditions for exposure, survival time, and pathogenic microbes in the environment. Providing recommendations for improving the environment by influencing its microbiota and assessing effectiveness of activities in this direction. Use of useful microorganism destructors, for cleaning the environment from harmful substances. Basic principles of regulation of microbial contamination of environmental objects as the present stage of development of microbiological science and methods of sanitary and microbiological research are as follows. First, absence of pathogens of bacterial and viral etiology in certain volumes and mass. Second, substantiation of macrobiological indicators and their permissible levels in which environmental objects are considered to be epidemically safe. Third, use as regulating indicators of sanitary indicative microorganisms. Indicator principle was and remains the leading one in assessing both bacterial and viral contamination. The need to use of sanitary indicative microorganisms is due to the difficulties that occur in the direct determination of pathogenic microorganisms. Their uneven distribution, complexity and insufficient effectiveness of existing methods for determining the number of all possible pathogens of bacterial and viral diseases. Sanitary indicative microorganisms (SIM) are microbes that are representatives of the normal microbiota of the human body and serve as indicators of the sanitary condition of the objects. Its contamination with human excretions. When choosing a sanitary indicative microorganisms, the indicator value of a particular microorganism relative to the corresponding pathogenic bacteria and viruses is taken into account, as well as degrees of development of methods for their determination. At the same time, it is necessary to consider the criteria of sanitary indicative microorganisms. Criteria of sanitary indicative microorganisms consist of 6 points. First, they must constantly be present in the body of man and animals and constantly excrete in the external environment. Second, they should not multiply in the external environment. Third, resistance and survival time should exceed those of pathogenic microorganisms. First, in the external environment, there should not be similar microorganisms. Fifth, they should not change in the external environment. And the last, they should be easily identified and differentiated. Indicator. When we tell about source of infection, we mean ill human and animal. But according to ways of microorganisms transmission, disease can be divided into some groups: waterborne, airborne, and others. Why? Because pathogenic microorganisms can certain period be reserved in water, soil. In a large segment of the world's population, the lack of sanitary water is responsible for billions of cases diarrheal illness that kills 2 million children each year. In the United States, nearly 1 million people develop waterborne illness every year. Through ordinary exposure to oil, soil, and effluents, surface waters usually acquire harmless, saprobic microorganisms. But along its course, water can also pick up pathogenic contaminants. Enteric bacteria are most useful in the routine monitoring of microbial pollution are coliforms and enteric streptococci, which survive in natural waters but do not multiply there. Finding them in high numbers thus implicates recent or high level of fecal contamination. Indices of sanitary microbiological quantity of water, soil, and air. The total microbial amount. The total quantity of microbes in 1 milliliter of water. The total quantity of microbes in 1 cubic meter of air. Fecal contamination, only for water and soil. The coli index, the number of coliform bacteria found in 1000 milliliters of water or in 1 gram of soil. The coli titer, volume of water in milliliters in which one individual coliform bacteria is present. The perfringens titer, weight of soil in gram in which one individual Clostridium perfringens is found out. Presence and quantity of indicator microorganisms. For air, Staphylococci and hemolytic streptococci. Presence of pathogenic microorganisms. Microbiological water monitoring is carried out with the safety and current sanitary surveillance. Safety control supervision is carried out in case of choosing water supply and the channeling of populated areas. The sanitary assessment of swimming pools, beaches, places of collective recreation. Current monitoring is carried out in case of first, assessing the quality of drinking water supply in populated areas. Second, assessing the sanitary state of surface waters in order to determine the degree of biological contamination effect on the processes of self-purification. Third, monitoring wastewater treatment. Last, epidemiological indications to find out the possible route of transmission of infectious diseases. Identify sanitary indicative and pathogenic microorganisms. Drinking water standards microbial contamination. Total microbe amount less than 100. Coli index not more than 3. Coli titer not less than 333 milliliters. Your recent lecture about microbial ecology, microbiome, clinical and sanitary microbiology. Lecturer Andrushkova Natalia Grigorievna. Thank for your attention.